Okay, so I'm going to be talking about, as Nushin said, I'm going to be talking about theta nested gamma oscillations. I think gamma oscillations in the hippocampus are really cool. There's some very interesting ideas about them. There's two different ranges. There's slow gamma and there's fast gamma. Slow gamma in CA1 is usually synced up to CA3. And I think the current ideology on that is that it's CA1 kind of accessing the stored representations or like memories in CA3. And fast gamma oscillations are what I'm going to focus on today. Usually they can get phase locked to the medial interneuronal cortex, which I think the current ideas on that is that's how it's accessing and like using sensory input. But another thing that I think is really cool and really important for what I want to talk about today, and is talked about a lot in this experimental paper, is that there's also fast gamma oscillations intrinsic to CA1. And the reason that I think that's really cool is that medial interneuronal cortex, it synapses on the distal apical tree of these CA1 neurons, pyramidal cells. And dendrites tend to kind of act like a low-pass filter. So I think it would be really hard to drive CA1 pyramidal cells into fast gamma if your inputs are that far away. But if there's already intrinsic fast gamma in CA1, then it might make more sense that instead of driving it, the medial interneuronal cortex fast gamma is syncing it up. So that's my goal today. My goal for the past couple of weeks was to try and model this intrinsic CA1 fast gamma. I'm sorry to say I was not successful, but these are the results that I do have. So this paper was really cool. It was looking at how could we investigate these different fast gammas. And what they chose to do is drive the pyramidal cells in each different area that they were interested in, optogenetically with a 5 Hertz sine wave. So the blue is that 5 Hertz sine wave, and they're driving it. So my goal, Nushin took the model and added another layer of detail by putting in her model the CA1 neuron and modeling the different changes in the axon. I went the opposite direction. My goal was just to model theta nested gamma intrinsic to CA1. So what I was actually hoping to do was to go down a couple of sizes and only have a couple of cells. There's different oscillatory mechanisms. A big goal in the lab is to model all of them and then model the inputs from the MEC and see if I can get them to sync up. This week, I was hoping to work on ping. Again, I don't have the best results to show you, but I can talk about ping. Ping, the P is the pyramidal neuron. It's an excitatory neuron, and it should be talking to the inhibitory cells. And what I'm kind of expecting to happen is that when I excite the pyramidal cell with this 5 Hertz depolarizing current that I'm saying should be equivalent to this optogenetic input, is that it should start exciting the inhibitory cell, which should fire a burst. And these inhibitory neurons, if they fire a burst, should cause the pyramidal cell to not fire. But as soon as that inhibitory burst and that inhibition ends, the pyramidal cell should start firing, have a little bit of a rebound firing, and that would cause, some fast firing in the fast gamma range that I'm looking for. Interneuron interneuron gamma oscillations, those don't require pyramidal neurons at all. Those are the inhibitory neurons talking to each other and inhibiting one, and then as soon as that inhibition ends, the second one should fire, and they just go back and forth on that. Coupled oscillators, I just really like the idea of coupled oscillators. I think they're really cool. When I was learning about coupled oscillators, I was learning about how they were first thought about as two pendulums that were used to keep time on a ship with the same beam. And what they found is, sometimes if your clock wasn't perfectly calibrated, you would have two different frequencies of your clock. But what they found is if you're having them on the same beam, they will eventually take the same frequency, but they'll be out of phase with each other. And so coupled oscillators, the idea would be that all of the neurons in my model at the crest of theta, which is this drive which I'm modeling as a drive that I'm giving to the pyramidal neurons, all the neurons should be excited at that crest, but because they're all connected to each other, they might all be clocks, but they're sharing the same beam, so they should start syncing up together. And I really like that idea because even if it's not what CA1 is doing, it might be what CA1 is doing with the medial and rhinocortex. So I just really like it as an idea. My goal this week, though, was to implement ping. So I instead of adding more detail to the model, what I really wanted to do was to simplify the model and take it down several times. Instead of having a couple of hundred neurons, I want 3. I want the pyramidal cell that I'm stimulating with that 5 hertz depolarizing current sinusoid. I want the fast spiking basket cells because what I'm hoping is that my pyramidal cell will stimulate my fast spiking basket cell, which will burst. And as soon as that burst ends, the pyramidal cell will start spiking. I also have these OLM neurons in there. And I think the OLM neurons will be important for replicating some of the experimental results I was talking about with this paper. This is the experimental result. There's a source and a sink, and they flip-flop during that slower theta. And OLM neurons make sense for spatially where that is. So that's why we chose to incorporate OLM neurons in this much simplified model. So the first thing that I did is, I took the model that Salvador had already imported into NetPyNE, and I took all of the other neurons out. That's all I did. I took all the other neurons out. I just left one pyramidal, one OLM, and one basket cell. And then I drove a 5 hertz wave into the soma of the pyramidal cell to see what would happen. This is just showing you what they look like. So this idea looks like that when I used Dr. Salvador's model and took it down. And, you know, when I first drove this wave in, I was actually really happy because it looked like I was already getting gamma. And this slow gamma should be in the 40 to 50 hertz range. Fast gamma should be 60 to 80. So it looked like it definitely looks like I've got fast gamma. The problem is that my basket cell wasn't actually firing at all. I was expecting to have an inhibitory burst and then some rebound firing. So I started playing with the weights, and again, I could get the basket cell to fire, and I can get the gamma in the range that I want, but what I can't figure out how to do is to get it to look like ping. This is just more of my results. I think modeling is supposed to show you where gaps in your knowledge is. And I think my problem is that I really need to understand these fast spiking basket cells a little bit more. Previously, my work had all been a single neuron model of CA1. So it's not what I wanted to present, but it's also not surprising that I need to study fast spiking basket cells a little bit more to be able to get this mechanism. So it showed a hole in my knowledge, and I have a lot more work to do. But ideally, what I want to do is model ping. Then I would move on to model this as a coupled oscillator where they should all kind of want to fire during the peak of theta, but by talking to each other, sync up. Ing would require a bigger network than 3 neurons because their neurons should be talking to each other, not necessarily the pyramidal cell. I should be able to take the pyramidal cell out and they would still have fast gamma. The last slide that I have is eventually I would like to try and replicate some of this cool experimental data with the sinks and the sources. If I were going to do that, I might want to move to some morphologically realistic models. And I can load those up into NetPyNE, which I thought was really cool. It was a lot easier than I thought it would be originally. And I can load them in. And NetPyNE does have support for calculating current source densities. And I thought that was really cool. But before I get there, I should probably figure out how to model ping and ing and all that cool fun stuff. But I was not successful this week.
I would actually say you were successful. So yeah, I wouldn't. Modeling is hard work, and there's a million dead ends you can go down. Some people get luckier than others and avoid dead ends, but it's largely chance. No, it looks like you made good progress. I think it's great. Questions?
I just want to say I agree with Joe, and it's modeling can definitely be a tricky art. So it sounds like you made progress and it looks good. Yeah. Especially for such a short time.
Yeah.
Yeah, but
usually
oh, sorry.
Sorry, I was going to say we usually take years to build the model. So
yeah. Yeah, Carol, I was just you may have mentioned this, but I was wondering if simply it was a limit of using just the 3 neurons that you couldn't get ping. You think that's the case?
I was thinking about that because I kept the connections between the pyramidal neurons, the pyramidal neuron, the fast spiking basket cell, and the Ohm cell, and the Ohm cell. I kept those, but there's a lot of recurrent excitability with the excitatory cells talking to the other excitatory cells, and I think it can be done because what I was expecting was to get my inhibitory neurons to burst. I think I need to play with the parameters more to get the burst I was looking for, but. Yeah, the goal was to start with a 3-neuron model, expand it to a larger population, and then expand it to a morphologically realistic one. I have a lot more work to do.
That'll be interesting. Thank you.
Yeah, it seems to me like you might want to do some singled cell optimization to see if you can get in this case, your waveform of the action potential is going to be very important, right? So the single cell will have a lot of effect. Anyway. All right, thank you very much, Nushin and Carol.